So I'm assuming everyone could see my screen, right? I was speaking for the longest while and I just realized that I was muted. Sorry about that. Yes, no. Hello? Oh, okay, cool. Just checking. So yeah, I'll start over because I was speaking for the last 2 minutes and I just realized that I was muted. But my name is David Frederick. Right now, I'm a senior medical lab assistant at Millennium Heights Medical Complex. I'm St. Lucian. You could tell from my accent, I hope. Qualifications, I just completed my bachelor's degree in biochemistry and biology, first class honors this year. And then I did that at the University of the West Indies in Trinidad. I have 2 years experience of facilitating STEM related workshops. But the workshops I've done before was mainly around digital literacy. I'm not really in this field, but I'm really happy to, encourage young people to get into this field. Um, One of my goals is to launch my own biotech company in the Caribbean and really pave the way for the next generation to get into the field of biology, biochemistry, and biotechnology. My passion lies in genetics, understanding the link between our genetic makeup and a lot of diseases. So in my presentation, you'll find a lot of my content stems around genetics and diseases, because my goal is to also become a genetic counselor in the future. So I'm really hoping that I could share some interesting facts with you today and that you come out of this presentation learning something new. So let's get into the presentation. So today we'll be doing, right now we'll be doing biotechnology in medicine. And we'll start with what exactly biotechnology is. As biotechnology is the use of biological systems, organisms, or derivatives to help to develop products and technologies that improve human health. In medicine, biotechnology has revolutionized diagnostic therapies and preventative measures, leading to better patient outcome and disease management. So we're going to start with some biopharmaceuticals. Biopharmaceuticals, also known as biologics, are drugs derived from living organisms. And there are many biopharmaceutical companies that you have probably heard of. For example, Pfizer, AstraZeneca, Genentech, Bayer. And I know that a lot of people would encounter these companies during the pandemic when we had to deal with the vaccines, and a lot of people had controversy over the vaccines and how they worked. I remember when Moderna released their mRNA vaccine, people thought that it would change their genome and whatnot. But these are companies that are responsible for the creation of many pharmaceutical products that can be grouped into therapeutic proteins, antibodies, and vaccines. So now I have a short interactive activity that we could try. So on your phones, you could go to menti.com. Let me just present this. All right, so you go to menti.com and you enter the code 14751513. It's right there. I could put it in the chat as well. So one moment. Sorry about that. I think I sent it to the admin. So let me just get that. Send it to the general chat. Okay. Yeah, so you go to this website, enter the code, and just name some pharmaceutical drugs that you might have encountered or that you know of the top of your head. Okay, so I saw someone said aspirin. Yeah, that's a good one. No? Okay. Well, we could move on. Let's get back to this. So this is, I just wanted to share the importance of plants when it comes to biotechnology. Um, in my experience, we really overlook plants, see that they're boring. But plants are really important when we develop these drugs. And I just included this table to show you some common drugs that are created from plants. Obviously, there is a way longer list, but stuff like aspirin, um, morphine, codeine, all of these are created from plants. I won't attempt to pronounce those plant names because I know I'll butcher them. And you could tell that they have various uses, so anti-tumor, anti anti-fertility, cardiovascular. So that just shows how important plants are. And it's important to understand that because I know from the Caribbean, something that we always encounter, especially with the older generation, is once they get ill, the first thing they will say is, drink some bush tea. But they don't really, and they usually have a stigma against the use of pharmaceutical products. However, what you need to explain to them is that the pharmaceutical products are made from extracts of plants. So we do use plants to create those pharmaceutical drugs. And sometimes the pharmaceuticals are better, but that's something that we will discuss later. So now we're going to move into diagnostic biotechnology. So diagnostic biotechnology involves using molecular techniques to detect and identify diseases more accurately. Examples of this application include genetic testing, biomarker analysis, advanced imaging technologies, which enable earlier diagnosis, and personalized treatment plans. The first one we're going to really get into is genetic testing. Genetic testing is a medical procedure that involves analyzing an individual DNA to detect changes or variations in the genes, chromosomes, or proteins. And there are an array of genetic tests right now. Um, you can do paternal testing, which is a common one that's been done for a while now. That's when you find out the father of a baby, for example. There's cancer screening, which is blowing up, sorry. Carrier screening, non-invasive prenatal testing, and pharmacokinetics. Um, so in the case of cancer screening, that's where individuals will do a genetic test to find out if they have mutations in certain genes that have been linked to cancer. And when you do these tests, they're not 100% accurate. So meaning that, but because you have that mutation in those genes, doesn't mean that you will develop the cancer. However, it just shows that there is a higher likelihood that you will develop the cancer. Um, so these tests are done for several reasons. So for example, like identifying genetic disorders, assessing the risk of developing certain diseases, determining carrier status for certain conditions, and something that's really blowing up in the US especially is gaining an insight into your ancestry and genetic traits. And some companies that you would probably heard of is Ancestry.com and 23andMe. These are um, companies that do ancestry tests. So you send a sample of your saliva, and then they'll tell you whether you 20% African, 20% Indian, and whatnot. Um, but in regards to identifying genetic disorders, there are some um, cases of some diseases, well, I'll say disorders instead, where certain mutations in genes would cause you to get like, we'll see in a subsequent slide some examples, but for example, sickle cell, when you have that mutation in the gene would lead to getting that disorder. So if you could screen, like in a prenatal screening as before you're born, you could find out if your child has that condition. So then you could take the necessary steps. Because you know, people with these disorders don't live a normal life like a healthy individual. So doing this genetic test is really helpful, especially right now for precision medicine, for example. So now we're just going to some genetic disorders. Um, I put three that I really wanted to touch on, three common ones. Cystic fibrosis, Tay-Sachs disease, sickle cell anemia. So sickle cell anemia is caused from a mutation in the HBB gene. And that mutation causes the production of abnormal hemoglobin and distorted red blood cells. So your red blood cell isn't the normal shape, and that would affect the amount of oxygen that you could carry. In the case of cystic fibrosis, there is a mutation in the CFTR gene, and that leads to the production of a thick, sticky mucus that affects the lungs and the digestive system. So to the right here, since I've done my degree in biochemistry, I've done a lot of pathways. Like once you do biochemistry, that's something that you would always encounter, and so that's something that you need to learn is understanding the pathways. So I just included a simple one here just to show you what the pathophysiology of cystic fibrosis looks like. So you start with the mutation in the CFTR gene, and I'm not sure if you guys have covered this at school, but when you have a mutation, well, genes are expressed to produce proteins. So if you have a mutation in the gene, then the protein that is produced would be faulty. So in the case of cystic fibrosis, this CFTR gene, when it's mutated, um, the protein that it codes normally is a carrier protein, well, a channel protein, which allows for the removal of chloride ions from inside the cell to outside. If you have a mutation in the gene, that protein is not coded for, and the chloride ions is not able to leave the cell. Now, when chloride leaves the cell, what happens is it draws water to itself, and that the water allows the mucus to thin out. But if the chloride ions cannot leave the cell, what happens is that the mucus begins to accumulate, and that's why you see here it leads to thick, sticky mucus that affects the lungs because there is no water being drawn to the area to thin it out. And when the mucus accumulates, it also leads to like it creates an environment for bacteria. So you find that you have a lot of bacteria in the area, and when you have bacteria in the area, you then get your immune system will then be triggered. So then you get a lot of inflammation, a lot of cytokines, and that's why these people have difficulty breathing because of that thick mucus that's in that environment, and also because of that inflammation. Um, but these proteins are also found on in the pancreas, so that's why you also have problems with the digestive system. So these are just some examples of genetic disorders. There are several out there. Um, yeah, there are several out there that exist, but in this case, it's simple because it's more for like one gene mutation. There are many disorders that encounter like you need multiple mutations to get the disorder, and that is what research is really being done on right now. Is like identifying what genes contribute to certain conditions, and if we could identify that, then we could look at therapeutic ways to change those mutations. And we'll talk about that more when we do gene editing and CRISPR-Cas9 in a subsequent lecture. But that is when we would speak about making edits to the genome. So if you have, for example, in the CFTR gene, we could change that mutation, then we could, lead to the person having a more normal life. But then we also speak about on the ethical side of it. But yeah, that's a discussion we'll have at a later point. All right, so this next slide, just to give you an insight of when I was speaking about the cancer, doing tests for cancer genes. So these are just different types of cancer that exist and the genes that are allocated to them. So for example, breast cancer, there's the BRCA1, BRCA2 genes. Now, like I said, if you do a genetic test and you find out that you have mutation in these genes, that does not mean that 100% you will develop breast cancer. That's not the case. However, what it means is that there is a higher likelihood of you developing the cancer, and that is when you would take the necessary steps. And we have an example of this called the Angelina Jolie effect. She's a famous actress. I'm sure you've heard about her. Her mom died from breast cancer, and when she did a genetic test, she found out that she had the mutation in the BRCA1 gene. So this gene right here. So she found out that she had a mutation in that gene, and what she did was she estimated that she had an 87% chance of developing breast cancer and a 50% chance of developing ovarian cancer. So let's go back to this. You can see that this gene is also for ovarian cancer as well. And so what she did is that she did a surgical procedure to remove both of her breasts to ensure that she does not develop the cancer. So again, it's not 100% that she would have gotten it. Sometimes she could have lived a healthy life and not developed any cancer. But since she realized that she had the mutation, she took the necessary steps to, for sure, not get that cancer. So that's a cool, and that's why they call it an Angelina Jolie effect. Let's just go back to this side, because people seeing that she did that procedure have now started to do genetic testing to also get some insight into their genome and find and take those necessary steps if they have to. Yeah, but that's it for genetic testing. It's a very interesting field to go into. That's why I want to go into genetic counseling. I feel like our genome really defines who we are, and I feel like it will really be a good career path. But now we move into biomarker analysis. So biomarkers are measurable biological indicators that provide valuable insights into various physiological and pathological processes within the body. Analyzing biomarkers has become an essential aspect of biotechnology in medicine because it enables early disease detection. You now have precise diagnosis being done, treatment monitoring, and the development of personalized therapies. So techniques in biomarker analysis include genomic analysis, so DNA sequencing, expression profiling, proteomic analysis, metabolic analysis, imaging techniques, and blood test biomarkers. One common one is blood test biomarkers. So again, my background in biochemistry led me to put in a slide like this. I don't expect you to understand everything that's going on there, but it was a really good example of how different markers could help you make diagnoses. And this one was a blood biomarkers for Alzheimer's disease and Down syndrome. So Down syndrome is another genetic disorder where the individual has an extra chromosome at um, at chromosome 21, and we call it trisomy. So because of that extra chromosome, you have an overexpression of genes. So that's one way. So if you did a genetic, so let's go back here. If you did a genomic analysis, you would see that that person has an extra chromosome. And then if you did you could see the blood biomarkers down here. That person has increased interleukin 6, 8, and 10. Then these are cytokines to show like the immune system is really working, and that is due to the immune dysregulation. So it was just an interesting pathway that I wanted to share with you. I could also share the article if you're interested in actually really know what this is about. But you don't need to live this. I don't expect you to live this. So precision science slash personalized medicine. Precision medicine is a cutting edge strategy that customizes disease treatment and prevention. The technique considers the patient's unique genetic and molecular composition, environment, and lifestyle to determine the route of treatment. So it empowers healthcare professionals and researchers to better predict the most effective treatments. So essentially, the same treatment does not work for everyone. And we could understand that from our genetic makeup. And I have a few examples that we'll go into. But first, I want to speak about what pharmacogenetics is. And this is a bit of a woody slide, I apologize. But pharmacogenetic testing is an integral part of personalized medicine, and it harnesses the power of genetics to tailor medical treatments for individuals. So this innovative approach considers an individual's genetic makeup to optimize drug effectiveness and minimize adverse reactions. So like I said a while ago, the same treatment does not necessarily work for everyone because the genetic variation of an individual can impact how drugs are metabolized, absorbed, and interact with other biological systems. And some examples of this is warfarin, that's like a blood thinner. Variations in certain genes impacts how individuals metabolize this blood thinning medication. So it would affect the dosage that you would give that individual. Additionally, we have coding, for example. Genetic insights reveal individuals who are poor metabolizers of codeine are at risk of adverse effects like respiratory depression. Now, we can't know that from just looking at an individual. Like I don't know, I can't look at you and know your genetic makeup. However, if we're doing a genetic test and we find out that information, then we can know, well, medical professionals would know how much should they recommend when they're making prescriptions. Okay, so that is the future. That's the future of pharmaceuticals. Um, and I also have some simple examples like asthma, for example. Asthma is a respiratory condition where cellular and molecular processes lead to airway inflammation, bronchial constriction, and increased mucus production. So, man, most people use aspirin. Like once you have some form of pain, you just consume some aspirin. You don't care if you have asthma or no asthma, you just drink it. But something that we should know is that aspirin could actually trigger attacks in asthmatic individuals. So when you take asthma, some asthmatic individuals could have like a reaction that would cause their airways to start constricting and they can't breathe properly, and that could lead to an attack. Yeah. So that's a simple example of the case. Like if you if you know that person is asthmatic, then you would not prescribe aspirin. Another one is cancer. And cancer is a condition where the cell cycle is disrupted and that leads to uncontrollable growth. So this can spread and affect other parts of the body. So all cancers are caused by gene changes of some kind. Doctors will utilize precision medicine to find out which individuals are high risk for cancer, discover certain cancers early, diagnose the types of cancers, should choose the best cancer treatment, and assess how well the treatment is doing. So now I'll touch on some, before I continue, does anyone have any questions? Any questions from the class before I continue? Because I know it's a lot of information. I was hoping to get some interactions and some questions along the way, because I know this is a very interesting part of biochemistry. But it doesn't look like I have any questions at the moment, so we'll continue. Right, so antibodies are small proteins naturally created by our immune cells to fight against pathogens. The mode of action of antibodies involve them sticking to specific proteins on the surface of cells known as antigens. So antibodies are created by your immune system. Oh, I have. Okay, thanks. So antibodies are produced by our immune system, in particular your B cells. So I guess we'll touch on that when we do immunology. But your B cells produce antibodies, and these antibodies are specific to different antigens. So like I said, we'll go into more detail about that when we do in immunology. But monoclonal antibodies are man-made antibodies that target specific proteins on the surface of cells. They are produced by cloning immune cells that generate identical antibodies. These precision targeting agents have revolutionized cancer treatment and are increasingly used in immunotherapy to treat autoimmune diseases. So researchers can design antibodies that specifically target a certain antigen, such as one found on cancer cells. So it's pretty straightforward. Once you understand how antibodies work, what they do is that they would bind onto, if you could see this image on the right hand side, the antibody has a heavy chain and a light chain. The light chain is where the antigen, where it recognizes the antigen, and you get like multiple of them swarming around a cell and binding onto them. And then what happens is after they bind onto it, you'd have your other immune cells like your phagocytes. So for example, your macrophages or your neutrophils, they would come in and they would eat up that complex of antibody and cells, and that is how you would destroy the cells. But again, like I said, we'll go into more detail when we talk about immunology. Yeah, so let's move on. So this is a better diagram of the mode of action. So as you can see on the right hand side, this is a target cell in blue, and the antigens are the yellow things sticking out, the yellow receptors sticking out. And what happens is the antibodies, which are in green, would surround the cell, and again, they would bind onto the antigen and make it easier for your phagocytes to now come in and swoop in. So this theory is particularly effective in the case of viruses. For a virus to enter a cell, its anti-receptor must bind onto the receptor found on the surface of a cell. So if you could design monoclonal antibodies that are specific to the anti-receptors of a virus, then you're able to basically impede their mode of action, like how they would infect the cell. So we'll do this in the next, I'll show more viruses when we do medical microbiology. But what happens is the virus has receptors, well, anti-receptors on their surface. And these are essentially your antigen. So if you could design monoclonal antibodies that can recognize those anti-receptors, then you're essentially preventing the virus from binding onto our cells, our host cells, and getting entry. So I had a brainstorming question down here. If this theory is so straightforward, then why are there viral infections? Why aren't monoclonal antibodies used to cure HIV, for example? So let's see if anyone could give me an answer as to why you can't use, well, you can use it, but why haven't we, stopped all viral infections? Any suggestions? Right. So yeah, Giovanni, that is a good answer. That is what I was looking for, mutations. Um, viruses viruses are so interesting. I did my last semester at university, I did a virology course, and you just learn so much on viruses. And yeah, that's the answer, mutations. So if we were to design monoclonal antibodies against viruses, what would happen is that eventually one virus, it would probably start working when we just introduced it, but eventually one virus would mutate and get like resistance to those. Well, they'll be able to change their receptor. So once they change their receptor, then the antibodies are more or less useless, because you need, for the monoclonal antibody to work, it has to be specific to that antigen. So once the virus receives some mutation that changes the antigen, right away it's useless. So you spend years developing that one thing, that one monoclonal antibody, just for it to be useless after one mutation. So that's why we don't really use it against, well, they're still trying to use it, but again, especially with viruses like HIV that rapidly mutates, it wouldn't be that effective. So yeah, great answer. So that concludes my presentation on medical biotechnology. I wanted to end with Cahoots. I'm not sure if any of you have played this game before. It's a very interactive way to recap the content. So in the case that you have not played it, I will just open it. Oh, I just saw some examples here. Peninsula and Sycamore. Yeah, I didn't see those. Yes, these are some good examples. So let me just open the Cahoots. I don't see many students, but it will still be fun for you to participate. So we'll do biotechnology in medicine. So one moment. What up? Okay, so on your phones or your laptop, you would go to, let me stop the music because it's kind of, yeah. So on your phones or laptop, you would go to kahoot.it. So I could copy this and put it in the chat. All right, so you go to kahoot.it and then you'll be prompted to put in a code. So you put in this code 5402495. So I'll give you some time to put it up, and then you put in um, your username. And it's a fun game. So the way you get points, in case you've never played before, the way you would get points is by answering a question correctly. So it's a multiple choice question. You have like 20 seconds. I put 20 seconds per question. And then the faster you answer the question, and the if you answer the question correctly, and the faster you answer the question, you get points. And then it ranks who has the most points. So I put 15 questions. The questions are related to the content we just covered. So we have Rajan in, I believe the other person is Giovanni. So we'll wait for Giovanni and then we'll start. Oh, what issues are you having? You could unmute your mic, by the way, if that's easier to communicate. But what issues are you encountering? Okay. One moment. I'll try it on my phone. So let me just see the prompts. Yeah, so once you go to the website, you should get like a direct screen to put in the code. So make sure you went to kahoot.it and not, because if you went to kahoot.com, it would bring you to the official website where you have to like create a quiz. But if you go to kahoots.it, You should get like the immediate prompt is to enter the code. It says game pin. Or what you can also do is you could scan the QR code. I could make it larger. Yeah, so you could use your phone and just scan this QR code and it would bring you directly to the page. Let's use the technology available to us. Let me try. Yeah. So you could use the QR code and it would bring you straight into the lobby. You just need to enter your nickname. Okay, so you're in. Okay, cool. Good. Okay, so we're going to start. So like I said, you get points by answering the question. Yeah, no problem. So you get points by one, answering the question correctly, and two, how fast you answer the question. So we're going to start. Right. So first question, what is the role of biopharmaceuticals in biotech in medicine? Is it developing new video games, producing therapeutic proteins and vaccines, improving transportation systems, or studying ancient fossils? So you have 4 seconds, 3, 2, 1.
I'm just seeing the shapes. I'm not seeing the question or the answers.
Yeah, so you don't need to see you just need to click on the shape. So look at my screen for the options and click on the respective shape. So triangle would be red. I think that's a diamond would be blue. The circle would be yellow and a square would be green. So you get that? So let's try it for the next question. So you don't need to, so look at my screen for the question, but just click on the answer on your device. So I guess because Rejani didn't take a question, Giovanni alone got it correct. So let's go into the next one. So the next question is, what are biomarkers? So is it bacteria used in medical treatments, measurable biological treatments, specific medications for genetic disorders, or biotech tools for cloning genes? 2, 1. Okay, so the answer was measurable biological treatments. So it's not specific medications for genetic disorders. It would more be something that we could measure to find out if someone has a different pathologies, for example. So right now, Giovanni is in the lead. So the next one, what role do biomarkers play in cancer diagnosis? Does it determine the blood type? Does it guide treatment choices? Does it measure bone density? Does it treat bacterial infections? So what role do biomarkers play? Yeah, so both of you got it correct. It does guide in treatment choices. Yeah. So right now you have to try to catch up, Risha, and you probably have to answer the question faster than Giovanni to get more points. So in which field do biomarkers assist in diagnosing heart attacks and heart failure? Is it cardiology, dermatology, astronomy, neurology? Yeah, so both of you are going correct, cardiology. Next one. What is the main focus of biotech in medicine? Does it improve crop yield, advance medical research and treatments, develop new smartphones? Okay, so I didn't have to read the prompts. But yeah, good job. You have to close the gap. So true or false? Personalized medicine eliminates the need for medical treatments. True or false? Does personalized medicine eliminate the need for medical treatments? Yes, that is false. Good job. Rajan is slowly closing the gap, by the way. Another true or false, aspirin can trigger an asthma attack. True or false? Can aspirin? Yes, that's correct. Next one. Cystic fibrosis arises from a mutation in which gene? So in which gene does cystic fibrosis arise from? Is it BFT, FTF, CFTR, BRT? Yeah, so the answer is CFTR. Someone said FTF. In case you weren't following, we did show, I did show it on the slide. So here it is the CFTR gene that codes for the not calcium, chloride, sorry, iron. So Giovanni is on a streak, the lead is opening again. True or false, Down syndrome is due to an individual having an extra chromosome at 21. So at chromosome 21, that person has an extra chromosome. Is that true or is that false? Yes, that is true. Next quiz, which one of the following is not a use of genetic testing? So is it to use genetic testing to identify genetic disorders, determine carrier status, ancestry tests, or protein count? Yes, so that's correct, protein count. Do a blood test for that. So next quiz, which one of the following is an application of genetic testing? So platelet count, PCR, pharmacogenetics, or urine composition. Which one of the following is an application of genetic testing? So which one of the following did you use genetic testing for, essentially? Yeah, so the answer is pharmacogenetics. So that is when you do a genetic test to find out which drugs work best for you. And we did that as in like an example of precision medicine. So far, Giovanni has gotten all 11 correct this far. 3 more questions to go. Asthma is a condition that causes what? Bronchodilation, bronchoconstriction, depletion in mucus, or sneezing. Yeah, so it's bronchoconstriction and not bronchodilation. So bronchoconstriction is where your airways become smaller, and that's usually triggered by some antigen. In the case we did today, it would be as aspirin. Bronchodilation is what you would use when you have an asthma attack. So you get something that relaxes your airway, so that way you could get more air passing through. So the answer was bronchoconstriction. Giovanni, sorry, got that correct. True or false? Monoclonal antibodies are man-made. Very simple one. Monoclonal antibodies are man-made. Yeah, so the answer is true. They are man-made. One before the last question. What is the primary goal of pharmacogenetics? Create genetically modified organisms, develop new recipes, customize drug treatments based on individual. Yeah, I didn't have time to finish, but yeah, that's correct. And last question. Which one of the following is not a biopharmaceutical company? BioGenTech, Moderna, or Zenith? Which one of the following is not a biopharmaceutical company? Yeah, so the answer is Zenith. That is the name of a country, I believe. Oh, I can't remember, but yeah, I just put based on my knowledge in football. But in second place, we have Rajan, and first place, we have, well, it has some drama effects, but it's Giovanni. He got all 15 questions correct. Okay, cool. But yeah, that concludes my presentation on biotechnology in medicine. So there's a lot more that we have to do, but we'll do that in subsequent sessions. So I'm not sure if I should jump into my next topic, or are we doing? I've read articles where persons are immune to HIV. Are they immune to all variants or some or one? Well, the thing with HIV is that it's one of the viruses that has several variants. So it is possible that a person is only immune to only one variant. It's very difficult to find that you immune to all variants of HIV. But that's something that I could get some more information on for you and share it. But in my experience with HIV is that like because of its genome, it could rapidly make changes to it, and then that makes it difficult for our immune system to recognize. Or it could just be that that person has some genetic makeup that makes their immune system superior. That is possible also. Um, but yeah, it's really rare for it to be fully immune to HIV. That is why we don't really have treatments at the moment. The most we have is some drugs that could lower the effects, but we don't really have a full treatment for HIV at the moment. Yeah, no problem. Yeah, but viruses are a really interesting topic. That's something that you should, if you're interested, you should look into doing virology. Um, so next question, I'm assuming. I'm not sure if Giovanni is typing. If I'm doing microbiology, would I do biotechnology? But the thing with, well, these are like you could literally do like careers in microbiology. You could become a microbiologist. You would use some stems of my biotechnology, like some procedures, some different techniques. So you could think of my biotechnology being, how would I say it? I'm trying to think of it, but like when you do microbiology, you do some parts of like of biotechnology. Like they kind of go hand in hand. But with microbiology, it is so vast. Like you could go into virology, you could go into bacteriology, you could go into fungi and study. You could study fungi, you could study parasites. So all these would fall under microbiology. Like it's so broad, and biotechnology is like techniques you would use to assist you with microbiology. I probably said it in a confusing way, but I really hope I answered your question. I feel it'd be easier if you. Okay, cool. Yeah, but you can do courses just on biotechnology where you learn like a vast number of techniques. You go into DNA and you do CRISPR, for example. All these would fit under biotechnology. And microbiology is when you study microbes, but there are different levels of microbes that you could study. So there's, like I said, viruses, bacteria, fungi, protozoa. Yeah. Okay, cool. So are we, I'm not sure if I should jump into medical microbiology or if we're doing constant fractions right now with Christine. So can someone let me know, please? Yeah, no problem. I try to keep it interested. Um, because like I said, I like genetics and diseases. So I could share the slides with you as well with the class so you could do your own reading. My advice is to go online and look at some articles and just read them. There's a lot of advancements being made right now in this field. I don't see anyone answering, so I think I'll just jump into medical microbiology. Yeah, so just to answer yours, just to go through some of the questions that you may have, Giovanni will do some medical microbiology. So many moments. Could I work at a hospital? Oh, you're doing the, oh, okay. Just, I wanted to find, okay, cool. Well, I'll stop sharing. But just to answer Giovanni's question before we start, yes, you can work at a hospital. Right now, I am at a hospital as a senior assistant, a senior medical lab assistant. So it's really what you want to become. But I'm just an assistant right now with my degree. I would still need to do further courses to become like a medical technologist where I could run departments like microbiology or serology. Yeah, but you can work in a hospital. It might be a little bit more difficult, but if you want to work in a lab, you could do that. And on a day-to-day, I assist medical technologists. So run different tests. So it depends on the department that I'm in. So if I'm in microbiology, what we would do is we would test urine samples. So from the patients in the hospital, they bring in the urine samples, we could test it for bacteria. We would plate the samples onto different agar plates to see which bacteria grows. And then you could also see what antibiotics they're resistant against. And then based on that, you would know, you could provide the doctor with the information and then they would be able to prescribe certain antibiotics. You could also, we would also visualize some of the samples under a microscope to look at like if you have different casts, because based on that information, you could like tell if someone has different pathologies. But that's just for that's just the microbiology part of it. So you'd also do the same thing for stool samples as well. Yeah, but like I said, depends on how long does this take you in regards to what the degree, the sampling. I'm not sure. Oh, the test. It doesn't take long, but it depends on the number of samples that you have. Sometimes you get like 10 samples in a batch or more, but it doesn't take long. Once you're used to doing the test, it's pretty simple. Like you put on your headphones and put on your gloves and you open your samples, and you take your samples, spread on a plate. It doesn't take long. But I think we should let Christine present. I'm open for questions after or in my sessions. Yeah.
I'm presenting on fractions and decimals. Fractions and decimals, they are actually quite simple to understand. Usually learn them pretty early on in probably about grade 3, and use them for all of math, even complicated calculus. And we're going to be learning how to add, subtract, multiply, and divide them today. Fractions are a way of representing parts of a whole. The top is called the numerator, and the number on the bottom is the denominator. Decimals are a way of representing the parts of a whole in base 10. The digits to the right of the decimal point represent tenths, hundreds, and so on. Equivalent fractions and decimals represent the same value, but are written differently. We learn how to convert between fractions and decimals and how to find equivalent fractions and decimals. Fractions and decimals are used in many real-world situations, such as cooking, measuring, and budgeting. So like when you read a cake box, the store-bought cake box, and they tell you to measure a quarter cup of oil or water. Yeah, so you use the liquid measuring cup or the dry measuring cup, and you see which one has the reading. And yeah, so that would be using it in cooking. Okay. Sorry. Okay, so when you break something into smaller pieces, you think of it as a fraction. So you think of the pizza and you get, most times it's equal pieces, around 12 slices, I believe. So when you take one out of that slice, or a cake, any circular food that you can think of, and you cut it into however many slices you want from it, you take one out of it, and that would be 1 out of 12, for example. Simple fractions like a half, a quarter, and a third, where we see here the 1 for the third would be the numerator, and 3 would be the denominator. They're easy to represent, and 1 piece out of 2, 4 or 3 equal parts. Adding and subtracting the fractions when the denominators are the same, like a quarter and a quarter, you add the numerators and keep the denominator the same. So for this one, it would be 2 out of 4, which would break down to just give you a half. And when the denominators are different, like a quarter and a third, you find a common denominator and add the numerators. So for 4 and 3, since we know that 4 and 3 can go into 12, we would use 12 as the denominator. And to find the numerator, we would divide 12 by 4 and multiply it by the numerator. So it would be 4 into 12, and then you get 3. So 3, and then 3 into 12 would be 4. So then it would be 4 plus 3 over 12, which would give you 7 over 12. So that's how you would add a quarter plus a third. The decimals are like fractions, but they use a dot instead of a fraction bar. So 0.5 is the same as 12. Is the same as 1, is the same as a half. Decimal places, the numbers are after the dot in a decimal, and they represent smaller parts. Like in 0.25, the 2 is in the tenths place. So you'd have 2 over 10. That's how you represent that. And the 5 is in the hundredths place. Decimal, the line up the decimal points when adding or subtracting decimals, and then solve just like whole numbers. So let's take 0.5. Okay. Can you see my cursor?
Yes, miss.
Oh, you take 0.25 and then let's add it to 0.5. Since the 5 would be in the tens place, you would just have 0. Okay. I can annotate. Okay. Since it would be like this. So then you would have 5, because any number plus 0 is the number. And then 2 plus 5 would give 7. 0.75. Yeah. Most times, well, the most common applications that you would see decimals apart from, math class would be when you're receiving change or you're paying for something, or if you've ever worked as a cashier, 0.5 or 50 cents. We don't really use cents in Jamaica, so that's not really something that we would see that often, but yeah. Okay, so I'm going to post the link in the group chat. Oh, sorry. Mm, uh, are you able to play the game? Okay. Rajani, are you able to play the game? Thank you.
Okay, so hope everyone is back. We could continue with our last session for the day. So we're going to be doing medical microbiology for the rest of the afternoon. Um, just checking if everyone is there. So can Giovanni and Rejan just send a message in the group to confirm that you guys are back? Okay. Okay, well, let's get into some medical microbiology. So medical microbiology, the unseen worlds of disease. Medical microbiology is a branch of microbiology that focuses on the study of microorganisms and well, their impact on human health. These microorganisms include bacteria, viruses, fungi, parasites, and understanding medical microbiology is crucial for diagnosing, treating, and preventing infectious disease that pose significant threats to human populations. So now we're just going to touch on like some types of microorganisms. So starting with bacteria. Bacteria are single-celled prokaryotic organisms that can cause diseases such as tuberculosis, strep throat, and urinary tract infections or UTI, very common in females. Viruses, on the other hand, are intracellular parasites composed of genetic material surrounded by a protein coat. And there are several examples of viruses. One major one is SARS-CoV-2, or we commonly know it as COVID-19. There's influenza, HIV, we go to herpes, which is HSV, hepatitis. There are so many viruses out there. Fungi are eukaryotic organisms responsible for fungal infections like candidiasis, and aphids foot. Parasites, eukaryotic organisms that live in or on a host, causing diseases such as malaria and hookworm infections. Yeah. So this slide just shows the exposure to pathogens. So there are different routes that a pathogen could enter your body. Your eye, your nose, your mouth, placenta in women, and the different genital areas, the broken skin through needles, and like insect bites. So for example, mosquitoes spreading dengue and the infamous chikungunya, Zika. Yeah. So entering a pathogen is known as exposure contact. A pathogen must gain entry into that's type of it, into its host in order to cause damage. um, the human body has several portals of entry. So I had this there to guess at first, but then I included this graphic instead, so that you could visualize the different like the different sites of entry into the body. Usually your skin is pretty protective against entry of these microorganisms. So that's why this one has broken skin, because that's when you get like a cut. That's why when you get a cut, the first thing they tell you to do is to wash it with hydrogen peroxide or rubbing alcohol to kill off any microbes that may have entered. Because if you leave it exposed, then more will enter and then you get an infection, I guess. Let's just go back. This to go up. So we'll start with pathogenic bacteria. These are responsible for a wide range of infectious diseases like pneumonia, for example, urinary tract infections, food poisoning, E. coli can lead to food poisoning and also UTIs, skin infections, so streptococcus pyogenes and staphylococcus aureus. And in particular food poisoning, we could, like a lot of people say, you shouldn't leave meat outside or just food outside because you could get bacteria starting to develop. Also, salmonella is another common one, that bacteria that grows on those food. But let's just look at the stages of a bacterial infection. It starts with incubation, where the bacteria has entered the body, but symptoms are not yet apparent. And then stage 2 is where you get mild symptoms or a vague feeling of discomfort that might start to appear. Illness is when the infection symptoms are at its peak, and convalescence, is when the immune system effectively fights the infection and symptoms begin to gradually subside. So it starts with incubation, that's when it gains entry. So there are different, like I showed you a while ago, the different routes of entry. It could be a cut, or it could be that you ingested, so you ate food that or something that contained the bacteria. And then you slowly start seeing some symptoms, and then you get illness. That's when you have like a peak of the bacteria growing inside you. But eventually, that's the beauty of our body, is that eventually our immune system will respond. And if you're healthy, you're not suffering from any immunodeficiency, then your immune system should be strong enough to recognize those bacteria and then eventually start killing them. And then that's when you start the symptoms starts subsiding. Or in some cases, you could be prescribed with some antibiotics as well that could assist your immune system with killing those pathogens. So this is a typical bacterial growth curve. So this is where you start with the lag phase, and then eventually the numbers start to um, multiply, and that's where you get the exponential phase. That's when you get a rapid replication of the bacteria cells. Because bacteria, like I said earlier, they're single celled. So when they go through binary fission and they split into two, you find that if you had 5, then in a few seconds you have 10, then you have 20, then you have 40, then it just keeps going and going and going. And then eventually you get to the peak. That's when, because yes, it could continue multiplying, but you have to keep in mind that there is a limit on the food supply, for example. So they can't just keep multiplying because they have to reach a point that if they exceed that point, then they will begin to die. So you do reach like a plateau phase, and then eventually you start getting death. But death is mainly due to our immune system starting to recognize them and killing them off, producing antibodies and different phagocytes that will eventually kill them off. So identifying bacteria, this is a very crucial um, aspect of microbiology because it enables us to understand their characteristics, behavior, the potential impacts on health and the environment. Several techniques and methods are employed for identifying bacteria. Those ranges from traditional methods to advanced biotechnological approaches. And some approaches that have been used is like microscopic examination. So where you know, for a fact that you have bacteria infecting a certain, I'm not sure, like an organism, and you put it on a plate and you just look at it under the microscope. And based on what you see, the morphological features, you could make inferences about the bacteria. So if it's circular, then you know it's a coccus, or if it's rod shaped, you know it's bacillus. And there are different types of things that you could see from just the morphology. But if you want to go into more detail, that's when you would do staining techniques. So that's when you perform a gram stain. And I have a short activity to show you how exactly we do a gram stain in the lab, especially in the microbiology lab. Let's go back. The other thing you can do is you could actually culture and grow the bacteria. That's when you put it on a plate, an agar plate. So you spread it on an agar plate. You put it in an incubator for a few hours, and then they'll grow on the plates. I'm sure you would have seen some pictures of that. A lot of people become artistic with their streaking, but normally streaking is usually standardized. I don't have a picture of it, unfortunately, but I'm sure you could just go online and look at it. But let's do this short activity. I could share the link. I'll share the link in the chat. So in your free time, you could also try it out. It's a really cool activity to get some virtual labs, that you could do online. But we're going to do it together in the interest of time. Right, so this is the Virtual STEMs website. So you could see that there are different modules that you could take part in. Very interactive. You could do bacteria sampling, testing for corn mold. You could do gram staining. You could use the microscope. If you did not get the opportunity to use a microscope like at school, you could do that. But today we'll be doing gram staining. So let's take a look
Gram staining.
at this. Yeah, so she reads out the captions. So we'll just go with listening to what she says and do it also. Okay, so let's start.
Hi, lab assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria. All yogurt has bacteria in it, since yogurt is made by adding specific bacteria to milk. These bacteria are beneficial to humans. Unfortunately, undesirable bacteria like Salmonella or E. coli can also end up in yogurt. These bacteria can make people very sick. This is what plain, good yogurt looks like. Notice the uniform texture and color. Now take a look at this yogurt. See how the whey and water are separating? Are those air bubbles forming near the bottom of the bottle? This yogurt looks suspicious, but to tell if it is contaminated, we need to get a much closer look. Let's collect yogurt samples and prepare them for viewing under the microscope by doing a gram stain. In the gram staining process, we will use different chemicals to stain bacteria that are in the yogurt. Then we'll look at them under the microscope. The bacteria will be stained different colors depending on what type they are. Bacteria that stain a pink or reddish color like this are called gram-negative, and their presence means the yogurt is contaminated. Bacteria that are used to make yogurt from milk stain purple. They look like this in a microscope and are called gram-positive. Pink bacteria are called gram-negative. We are going to gram stain samples of the yogurt so we can see what kinds of bacteria are present. All right, let's prepare our slides for gram staining. To prevent bacteria from contaminating our work area, we have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loops we'll use later and help maintain a... You're going to use a striker to light the Bunsen burner. The gas is turned on low already. We need to dilute the yogurt before we put it on the microscope slide. We'll use a PBS liquid, phosphate buffered saline, as the diluent to thin out the yogurt. That helps us see the bacteria with the microscope. Start by placing 1 milliliter of the diluent into a test tube using a pipette. This is an inoculation loop, a small loop of wire used to collect samples. We'll use it to place a small amount of yogurt into the test tube with the diluent.
So this is a very accurate representation of what the inoculation loop looks like. And if you go into microbiology, that is something that you will commonly be in use of for just transferring samples from one plate to another or from a test tube onto a plate. This is also what you use to streak your plates. In the lab, what we have is some plastic inoculation loops. So you would use it once and throw it afterwards for when you're taking samples, for example, from urine or from stool. But in the case of research laboratories or even at university laboratories, you would have a wire loop. So you can't throw that one. So you have to continuously sterilize it. So you'll see how we sterilize inoculation loops in a bit.
Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot.
Yeah, so this is pretty accurate. So you would have the inoculation loop in your hand, and what you do is you put it into the flame and you keep it there until it gets red. Once it's red, that means that everything
After
under
it
is
has
dead.
cooled down, use the loop to obtain a small sample of yogurt from the suspect container. To get a good sample, reach deep into the container or thoroughly mix the yogurt first.
Yeah, so in this case, we're working with yogurt. So we get a sample of the yogurt to identify the bacteria in there. I'm speaking now, so I'm taking time to do it, but in the laboratory setting, after you sterilize your loop, it would be more beneficial to take your sample quickly, because just keeping it out here could contaminate it and screw your results. So you would take your loop, dip it in, and then you have your sample. So now you have your sample there.
Place the yogurt sample into the previously measured diluent. Now, gently shake the test tube for 5 to 10 seconds to thoroughly mix the yogurt sample throughout the diluent. Use the inoculation loop to transfer a small amount of sample from the test tube to your prepared slide.
So yeah, so now that we have it, we have to go back and take a sample from the small test tube we had and put it onto a slide. Again, we have to sterilize our loop because we transfer in we get a new sample. So we do the same thing. We go back to the, continue. Yeah, we're going back to
Sterilize the inoculating loop again in the
inoculation.
flame of the Bunsen burner. Hold it directly. Transfer a small amount of sample from the test tube to your place 2 loops full from
So
the
then we
sample
go in
onto
and
the slide.
put it on our slide.
Tilt the slide to spread the dropout slightly, about the size of. Now we'll heat fix the slide. This prevents the bacteria from being washed away when we begin the staining process.
So when we begin the staining process, we will be using liquids because it's a dye. So it's a liquid. So you don't want to put on your dye, and also when you put the dye, you have to wash it out. So we don't want to put the dye and we basically wash away the bacteria that is in our sample. So what we would do is we would heat up the slide, as you were saying, to fix the bacteria onto the glass. So that way when we apply our liquid dye, it does not wash away.
Place the slide in a glass, such as a clothespin, and pass it over the flame of the Bunsen burner. Now we begin the staining process. After the staining is complete, we can look at these slides using a microscope. We use 4 chemicals to stain a slide. We apply them in a specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze, let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of Gram's iodine. Gram's iodine will fix the dye to the cell walls of the bacteria and make it permanent. Squeeze several drops onto the sample, enough to cover it completely. Let that sit for 60 seconds. Now, rinse it under slow running water. Now we are going to use the alcohol solution. This will wash off the excess dye, leaving only the dye fixed to the bacteria. Use the alcohol dropper to cover the sample in alcohol. Let the alcohol sit for 30 seconds. Time is very important in this step. Don't let the alcohol sit for more than 30 seconds. Now, rinse it off under slow running water. You now have stained the gram-positive bacteria purple. The next and final step will stain the remaining bacteria pink. These bacteria are gram-negative. Use the dropper to cover the slide with safranin. Let that sit for 60 seconds. Now, rinse it under slow running water. Now our slide is ready, but it is still wet. To dry it off, place it between 2 sheets of highly absorbent bibulous paper. Gently press the paper down a few times to blot the water off the slide. Good job. You now have one slide ready to look at under the microscope. Try doing the next one on your own. You can always tap on the hint button if you notice the timer and the two buttons next to it. These buttons let you decide how long to wait for a stain before rinsing.
Okay, so we're going to do one on our own.
Remember, you can tap on the
We're not going to use the hint. So can you inform me of what the first step is? If you remember correctly. Let's see if you remember. So yeah, we started with the violet, correct? So we put
using
the
the dropper.
violets,
Squeeze out enough drops to cover your
and then what do we do now?
Let it stay for 60 minutes.
60 minutes. We're leaving it for an entire hour. Wait,
I mean seconds.
yeah, 60 seconds, correct. Okay, and what next? Yeah, we rinse it off. And next up, yeah, we add the gram iodine.
using the dropper. Squeeze out enough drops to cover your sample.
And what next? Yeah, we do a 60-second wait. Then, yeah, we rinse it off. Next step. Yeah, we add A.
Using the dropper. Squeeze out enough drops to cover your.
And how long do we leave it for? Yes, so this one is 30 seconds. And then we rinse. And then last step would be, well, the last step pretty obvious, but yeah, we add the saffron in.
Using the dropper. Squeeze out enough drops to
And
cover
how
your.
long do we leave this one for? 60 seconds, yeah. And then we rinse.
Good job. You completed gram staining a slide on your own.
Yeah, so this is how we do it in a medical laboratory. The reagents are lined up in this way to make it easy to not skip a step, because it would affect the entire results obtained. So you usually keep the reagents in this order. So let's continue. One thing I'm not sure if you guys have ever done the theory behind this, but just to give you a brief introduction into it, different bacteria, gram negative and gram positive, they're different because of their cell wall. You find that gram positive bacteria have an extra cell wall, no, not extra cell wall, extra layer, sorry, and that is what retains the violet color. So when you apply this violet color and then you add the iodine, both gram positive and gram negative bacteria at that point will be violet. But when you add the alcohol, the gram negative bacteria, they don't hold the violet because they don't have that extra layer, right? So that's why it washes off. So after it washes off, you would add the saffron in to then stain them. So that is the theory behind it. Obviously, you could read more on it or watch some videos on it, but that's just a brief introduction into the theory.
Congratulations. Here's what we found under the microscope. The image on the left shows only the desirable type of gram-positive bacteria and no sign of gram-negative bacteria. The one on the right is contaminated. Notice the pink-stained bacteria. Those are gram-negative. These contaminants include undesirable bacteria that could be harmful.
Yeah, so that just shows the application of this test. Well, in this case, we were doing yogurt. So when we're doing yogurt, you in the yogurt industry, you'd mainly use gram-positive bacteria. But in this case, you could tell that we do have some contamination because we have the gram-negative shown in our sample. So yeah.
Look at all those gram-negative bacteria. It's a good thing we caught this before the product shipped. Now we will need to do some investigating to determine how the yogurt became contaminated. and identify the exact strain of bacteria that contaminated the yogurt.
Yeah, so
You have completed the gram staining.
Yeah, so that concludes the short experiment. Just to show you how it looks in a laboratory. Obviously, I send the link in the group chat, well, in the chat, sorry. So you could take a look at the different tests available. Nice virtual labs if you've never done them at school, or if you're studying and you just need some like a refresher on those concepts. But yeah, so this is what the procedure would usually look like. We said fixation, so that's when we heat it up. We add our dye, then we add our iodine, then we decolorize. Like I was saying, when we decolorize, the gram-negative bacteria, they don't, let's just move that. The gram-negative bacteria, they don't keep the dye, so they'll become white. And then when we add the saffron in, they become pink. Yeah, so that's just the theory behind it. 1 moment. Okay, cool. So this is just a table showing you some common gram-positive versus gram-negative bacteria. So gram-positive would include stuff like staph bacteria, anthrax, enterococcus, group A streptococcus, and gram-negative would include E. coli, salmonella, gonorrhea, and H. pylori. So just some general information that you could take home along with you today. A typical bacterial infection can cause a wide range of symptoms depending on the affected site. But some common symptoms include fever and chills, pain, redness, swelling at the infection, coughing, and difficulty breathing in respiratory infections, nausea, vomiting, diarrhea, urinary frequency, and pain when you have a UTI, for example. So now we go into the interesting microbe to me, and that is viral infections. Viral infections are caused by tiny particles that can have a significant impact on health. And these are some common viruses. So SARS-CoV-2, also known as COVID-19, that caused the pandemic, 3 years ago, that kept us all inside and social distancing and really changed the whole landscape on a global scale. Then there's influenza, which is also known as the common cold, something that's been there for years now. HSV, these are more sexually transmitted diseases. So HSV is herpes, is HIV, hepatitis B, and hepatitis C. And these are what like the structures of some viruses look like. That's the papilloma virus, the rhinovirus, rotavirus. These look like these. This is the infamous COVID-19 virus or coronavirus. Yes, Rajan?
Why does the papilloma virus look so identical to the Mastinella virus and also the hepatitis B?
It looks identical, but it's not. These are just because if you look at the hepatitis B, for example, it has an envelope on the outside, while this one is a naked virus, so it does not have the envelope. This one also has different receptors, but the thing with viruses is that a virus is a very simple organism. It is made up of some proteins, like 3 proteins that are just repeated. I think I have a better image somewhere. I don't have it here. Oh no, I don't have it. I thought I had it, but I don't have it. Let's see if I could get a better picture to show you. But essentially, oh, tell me I had a picture of it. I could swear I had a picture. Yeah, right there. Yeah, I know I had a picture. So if you look at this virus, you can see that the colors are repeated. So like the structure of the virus, the capsid on the outside is made up of 3 proteins usually, and these 3 proteins are repeated. So you have, and what they do is like they come together and they form this shape on the outside, because a virus is very small and its genome is very small. So it cannot code for, like, for example, we have a larger genome because we need to code for so many different proteins. But because the virus's genome is so tiny, it could only code for a limited number of proteins. So what it does is it would code for 3 proteins and they would come together in an identical, like in a symmetrical way. And like that repeated unit like binds to each other. So you find a lot of them would have like, if they're related. So in this diagram, you see the papillomavirus, enterovirus, rhinovirus, rotavirus. If they're related, you find that sometimes they have like a similar shape. They're not the same virus. They, trust me, they would have different receptors, but they would have like a similar shape because they're so simple, but yet complex. Viruses are really interesting, trust me. But I hope that answered your question. If you go into virology, you would learn more about how they're formed. But just keep in mind, like I said, they have a small genome, so they can't code for a lot of proteins. So you find that what they would do is like they would create subunits. So 3 subunits that would form a shape. Like I try to think of like a building block. So think of a triangle, but then you repeat the triangle multiple times and like it fits together with identical symmetry and interactions in between them. And that forms a, well, viruses are known as having a metastable state, meaning they're both stable and unstable at the same time. So I hope I didn't confuse you.
So to identify a virus, you look at the pattern of the proteins and the
But
shape.
to identify, the best thing to look at is their genome, because it would be specific to that virus. It's not just the shape. Yeah. So you look at the genome. That's when we would use PCR, for example, to identify if someone had COVID, because the genome would be specific to that virus. It wouldn't be shared with another one. Yeah. So if you have any other questions, feel free to ask. But these are just some diagrams to show like the different shapes of viruses. And this slide, this slide shows the 7 steps in the replication of a virus. Now, I had to learn this for my virology exam last semester. But I just wanted to show you what it looks like because it is so interesting on how they move around in our bodies. So this is where it starts, where the virus binds to a receptor on the surface of a cell. There are different ways that it could gain entry. In this case, it gains entry through endocytosis, and then it's released. It's traveled along different microtubules. It binds onto, this is the nucleus. So it binds onto the nuclear pore complex and it gains entry where where in our nucleus, it would then hijack different systems to replicate, and then it bursts open and spreads to different cells. I just show you to show you the beauty of how viruses move along in our body and how they replicate. But yeah, not all viruses uses this same pathway. Some does not need to go into the nucleus to replicate. They could literally replicate in the cytoplasm of a cell. But something you need to keep in mind is that all viruses must use the machinery of its host to replicate, because on their own, they cannot replicate. They need a host. That's why we call them, in some ways, parasites. And this is a typical growth curve of a virus. It looks very different from a bacterial growth curve. So if you want to go back to just look at this is what the bacterial growth curve look like, but this is what the viral growth curve looks like. So you see, like, this point is when you have the inoculation. So this is when the virus would bind to the receptors of the cell, and you would find that when it enters the cell initially, you would have an eclipse phase, because if you do a test, you you would not get any virus because they're in the cells, they're not in circulation. So most times, this is when you have an eclipse phase. Now, depending on the virus, you would have sometimes a short eclipse phase where they enter and they could replicate so quickly and distribute that the eclipse phase is almost like non-significant. But in some cases, you could have a very long eclipse phase, for example, with herpes. Yeah, herpes is like a latent virus. It stays in our nervous tissue for a long while before it, there's a burst phase where there is a release of viral particles, and then eventually there will become an equilibrium, which is its limit. And then eventually you get a decline when our immune system starts killing them off. But yeah, it's very different from a bacterial infection. Bacteria does not really enter our cells, so you would never see an eclipse phase for bacteria because they're like just in circulation, they're just multiplying. But with viruses, they need to enter the cell, like I said, to replicate and then spread. Yeah, so just some cool information. Now we're just going to speak about some common viruses I wanted to share some information on. The first one is influenza, because that's something that I believe everyone has experienced in their life. So this is what a typical influenza virus looks like. Its genome has 7 segments. This one shows 8 for some reason, but it's 7 segments. And the key thing about influenza is they have 2 receptors that assist them to spread, hemagglutinin and neuraminidase. Somebody speaking? If you have a question, you could feel free to interject. That's not a problem.
It was a mistake.
Oh, okay, cool. Well, yeah, influenza, the 2 main receptors is HA and NA. These are found on the surface of the virus. Like I said, viruses need to recognize a receptor. In the case of influenza, the receptor is sialic acid, and that is one that is commonly found on literally all cells. Most cells have sialic acid. So they bind onto sialic acid, and then they gain entry into the cell. And because of the different segments, that is why you get like different variations of the flu. That's why we don't really have a vaccine for the flu, because it's very easy for it to replicate. Let me see if I could go back to this slide here. So let's see. So if we look at this point here to the top, this is where the virus basically breaks down and releases its genetic information. So you could realize that if influenza releases its 7, let's say this person was affected by 2 strains of influenza. So then when influenza is in the nucleus, it releases its genome. So right now, 2 strains of influenza, we have like 14 different strands, well, different strands of genetic information in the nucleus. And when it's repackaging, you could find that you could have like a mix of those genetic information, and that is how you would get a mutation with influenza. And if you look at the different strains of influenza, I'm sure you've heard of H1N1, H3N2. There are so many different strains of it because of that ability to mutate. And that's mainly because of its genome being able to, if you're infected by 2 different strains, it's very easy when it's repackaging to take some genetic material from a different strain and then you form a new strain. Yeah. So yeah, that's just influenza. Pretty interesting. And that is why we're stuck with not having like a cure for the flu. And that's also why you get multiple, like you get infected by the flu all the time. And some people will ask you like, okay, so I thought I got infected with the flu in January. I thought I was supposed to be immune to it, like my antibody, like I should have antibodies against it. But the thing is, you could get infected with another strain of the flu in, let's say, July, and your body would not have antibodies for that one because it's a new strain. So now you have to recover from that one and, form antibodies for it. So it's very interesting how different strains could cause the same effect. But yeah, so that's influenza. The other one I wanted to speak about is HIV, because this is another interesting one, in my opinion. Again, we don't have I say this, we don't have a vaccine for it as yet. But the reason why I put HIV is because it is one of the viruses that affects your immune system directly. So it recognizes, so if you look here, it recognizes the CD4 receptor found on the surface of T cells. So if you do not know, we'll touch it in immunology, but T cells are like your other form of like your immune system, that is your adaptive immune system. So these viruses would bind onto the surface of this CD4 receptor and then gain entry into your T cell. And if it's destroying your T cell, then you become immunocompromised because you're no longer able to, effectively um, destroy other pathogens. So are viruses naturally occurring? Yes, they are. They are naturally occurring. I was like, when I was preparing this slides, I was thinking like, what is the purpose of viruses? Like, I can't think of what good thing viruses do, because they are not part of the five kingdoms. Well, yeah, that's a big debate in the science community, really asking whether viruses are alive or dead. Some people believe that they're alive, some people believe that they're dead. My opinion is that they're dead because they require a host, like they cannot live on their own. They don't, there are several levels of like things living organisms do that they don't follow. Like yeah, they reproduce, they mutate, but at the same time, they don't meet a lot of criteria. So from my perspective, they're not alive, but you'd find people arguing that they are alive. But yeah, that's it for HIV. Like I said, if you're affecting T cells, then you affecting your immune system, and that's why people with HIV eventually develop AIDS, which is when yes, yeah, they really weird. And I wouldn't say weird, I would say they're really interesting. They're really interesting. But yeah, that's why people develop AIDS, because you're now depleting your T cells. So you find people who suffer from AIDS would like they would get any infection. So if there's a flu going around, you would catch it like what you'd be the first person to catch it because your immune system is not performing at the optimum level. Just be one moment. Yeah, so sorry about that. And this virus, HIV, another thing with it is that it is very, it replicates a lot and it also mutates a lot. So it's very difficult for us to design vaccines against it. And the fact that it targets your immune cells also makes it a big issue. But we have been able to develop some vaccines, not to cure you from HIV, but to prevent its spread. Just one moment. Yeah, so in the case of the antibody, antivirals that they've designed, it's more like a protease inhibitor. So that's really good into the biochemistry. I don't want to confuse you guys, but in something that's important for the virus to become mature is it has a protease which has to cleave some proteins within the virus. So once it does that, it is able to become mature and able to affect other cells. So what scientists have done is they've designed some protease inhibitors. So it inhibits the activity of the protease, and essentially the virus is unable to become mature and spread. That's why some people taking those antivirals are able to interact with other individuals and not spread the virus. But that does not mean that they've fully cured of it. It's still there. It's just that it's not active. So yeah, so I'm hoping eventually we'll get to the point where we can design a fully effective vaccine against it. But yeah, so that's, I personally believe that if they started creating vaccines early, we would minimize HIV, just like COVID-19. Biotech companies started to work on. Yeah, but the thing with COVID-19 is that it was very similar to other, there are very similar viruses in its family, like MERS, for example, and there's also another other coronaviruses out there. So the reason why we released vaccines so quickly is because like there was already a blueprint for them essentially. And also you had to think about the fact that HIV came out in a time where technologically we weren't as advanced. That's why it spread so much. It was difficult to spread education on HIV. Um, but I'm hoping that in the next 5 to 10 years, that with the advancement of technology and you know all those things we're doing with our genome, we're able to get some level of vaccination. But yeah, good point. Good point. So now we do go into SARS-CoV-2 or COVID-19. This is what it looks like. I just wanted to show this interesting fact. So like the receptor for SARS-CoV-2 is the ACE2 receptor. Now the thing with this receptor is that it is found on practically all epithelial surfaces. So it's found on several organs. And that is why when you contracted COVID-19, you found that there were a lot of adverse effects, like you got a lot of different effects. And if you look at just the different scientific papers out there, you realize that so many different things happen when people got COVID-19 and people, it wasn't just a respiratory virus, it was like affecting all levels. Like you could get Guillain-Barré syndrome, you get increased heart failure just because of the fact that its receptor is found on so many different cells in our body. That's it was able to spread. And yeah, if you're interested, you could and you're free, just take a read through the different articles showing like just the adverse effects of COVID-19. Very interesting. But that's it for viruses. Like I said, there's so much about viruses. I could literally sit and just speak about it. But I won't. I didn't want to put too much information on it. I feel like if you want to speak more about it, you could reach out to me, or I could share some articles that you could have a read and see. But now we move into fungal infections. So fungal infections, also known as mycosis, can affect various parts of the body. The study of fungi is mycology. Some fungal diseases is candidiasis. I think I butchered that. But it's a common fungal infection that can affect the mouth or genital areas. There's also athlete's foot. I'm sure like if you're very athletic, that's something that you may have gotten over the years and you buy a powder to stop it. But it's caused by the dermatophytes, which lead to itchy, flaky feet. And then also aspergillosis, which is an invasive fungal infection that primarily affects immunocompromised individuals. And this is a video that I wanted to show. It's from a Nat Geo about a fungus called the cordyceps. I'm not sure if you guys have ever encountered that, but it's a very interesting fungi. And it would lead to a discussion I want to have with you in the subsequent slides. So let's just watch this. I think it's about 5 minutes. Yeah, 4 minutes.
Fungi and slime molds race to decompose dead matter on the forest floor. Many spread by releasing spores, up to 30,000 a second. If just one of these spores lands in the right place and takes root, it can colonize a whole new area. But not all fungi feed on the dead. Days ago, a spore landed on this ant. Now she's acting strange. A network of roots has infiltrated her muscles. Her body has been taken over by cordyceps, a parasitic fungus. It floods her brain with chemicals, just the right amount of light, just the right amount of humidity for the parasite growing inside. It forces her to clamp down in a death fight, and cordyceps reveals its gruesome nature. After 3 weeks of growth, cordyceps can release its own spores, infecting more ants, releasing more spores, infecting more ants, releasing more spores, infecting more ants, infecting more ants, more ants, more ants, more ants. Cordyceps can wipe out entire ant colonies. But more than just ants are at risk. There are over 600 species of cordyceps spread across the world. Most are found in jungles, where they prey on a whole host of victims.
So yeah, this was a very interesting fungi or fungus that I wanted to share with you. I'm not sure if you've ever heard about it. Sorry, it started raining on my side, so it's pretty loud. I can actually get hear me properly still. Um, so apologies for that. But in regards to the cordyceps, I'm not sure if any of you have seen the show. Let's just go to the next slide. I'm not sure if any of you have seen the show The Last of Us or played the game. Um, it's a show that shows like a zombie apocalypse, um, almost like The Walking Dead. But this one is a bit more, I would say, biochemically accurate compared to The Walking Dead. And these are things that I pick up on when I look at shows. But there's a theory. The theory in the show is like because of climate change, the cordyceps, the fungus, is now able to infect um, humans. So right now, the cordyceps are not able to infect us because of the temperature of our body. Like it is so hot that it's like a hostile environment for them. So if we did ingest one of the spores, for example, it would like immediately die. But the theory of the Last of Us is that because of climate change, they've now adapted to the increased body temperature. And now we were to if we were to ingest one of the spores, then they would be able to brainwash us and eventually start like a zombie apocalypse. Because that's what they're doing with the insects. They call zombie ants. Because what happens is that when they're infected by the fungus, they move like they climb to high areas. That's what you were seeing in the video. Like they would drop everything that they were doing and instead climb to high areas. They would pinch like the mouse into a branch, like just hook onto the branch and then eventually die. And what would happen next is the fungus would consume all the nutrients within them. And because of the height that they're at, it would then spread spores to affect even more ants. So that is just an interesting thing about if they were to infect us humans, if it could start a zombie apocalypse. I don't believe so because we have other barriers other than heat. For example, the pH of our stomach would make it very difficult, and also our immune system is way more complicated than the ants. But yeah, that's just something to think about with the everyday realizing how much climate change is affecting us. Um, But yeah, that concludes what I had for today in regards to medical microbiology. I'm not sure if you guys have any more questions. We could entertain some questions. I have one more Kahoot that we could do with for today, but in regards to time, I'm not sure if we should do that in our other session. But I'll entertain any questions. So yeah, we'll do that in our other session, but I'll entertain any questions if you have any right now. Doesn't look like you guys have any questions. One more thing I want to talk about is a STEM experiment. So this year, what we'll be doing is an experiment where you would be extracting saliva, well, DNA, sorry, from your saliva. Very cool. I did it myself 2 years ago, and you would be able to visualize your DNA. I have a picture here on what it looks like. So it depends on how much DNA you extract from your mouse, which show. But this is what it will look like, more like a web. But we'll go into that tomorrow. And I'll send the materials that you need in the classroom so you could get that sorted and then we could try the experiment ourselves. I also have a few questions because you guys will be, I'm sure you will be thinking of going into university and getting into research in this field. So I have a few questions that you would answer. 8 questions. It doesn't have to be as detailed um, as I would answer them, but yeah. But that concludes everything that I have for today. Now, in other session, we'll do the codes.